I shaped somewhat my talk to what is in my interest. I'm also not a microbiologist, so I shifted a bit towards some more known territory for me. I must say that the task of this title is a challenge, especially after the time pressure Jazz was on. My gut microbiota are already being dysbiotic now, and on the other hand, I should also take this assignment in 20 minutes. So I will not go into depth because Jazz already alluded to this. So the clinical relevance of the gut microbiota host interaction is in fact that it is the backbone or the basis for the gut-liver axis and everything that follows from it. So this was already discussed by Jazz, so I'll skip this in the sake of time. And in fact, for the sake of my talk, I divided this lecture into 4 topics. So gut microbiota and host interaction in health and disease, very briefly. And then the potential of FXR, and maybe this brings us somewhat to the last question where there is a relevance towards bile acids and how they might influence permeability to bacterial translocation and microbiota. And then to end up with one slide, Mr. Chairman, on clinical applicability and finally some conclusions. So if we go to the gut microbiota host interaction at the basic level, then you end up in fact with a triad of gut flora, intestinal barrier, and the immune response, the local immune response. So if we start off with gut flora, the oldest paradigm, in fact, the first paradigm, was that all microorganisms are pathogens, are aggressors. And so in this view, the immune system was to go to war against these creatures, and the intestinal barrier was to hold them at the level of the intestinal lumen. But we have evolved, and in fact, we have evolved in our vision because we've learned that in fact bacteria are much older than us, and so probably more evolved than we are. So bacteria were already here 4 billion years ago, so before we came. So we should respect our elders, that's what I thought. So in that aspect, I think we should also respect bacteria. Moreover, if you look at us as a being, then we are in fact more microbes than we are human. I don't want you to go home afraid, but that's how it is. And so in that way, you should also know that in fact 90% of all genes are bacterial genes. And this has in fact led to the perception or the idea that we are superorganisms. We and our microbes, we in fact are a community in which complex transgenomic biochemical interactions occur in the advantage of both, so trophic, protective, and metabolic. And this in fact has led to the updated paradigm that we have tummy buddies, so good bacteria, and tummy baddies, bad bacteria, which in fact balance each other. And moreover, they are also balanced by the immune system that has needed to evolve to tolerate, to accommodate colonization of those good tummy buddies, and has to learn to search and destroy the tummy baddies. And to go in further depth about the tummy buddies, I mean, these are really our best friends. So reconsider the concept of best friend. These are your bacteria. They are very important, and Jazz already alluded to this, because they are prohibitive for pathogen colonization due to direct interaction, because they alter the environment, which make pathogens live less valid. They consume the nutrients that are required by pathogens, and they produce specific metabolites which might affect pathogenic virulence, amongst others short-chain fatty acids. They also alter conditions required for virulence activity, amongst others the ambient oxygen tension. On the other hand, they also indirectly prohibit pathogen colonization by interaction with host immunity. And every qualitative or quantitative change in composition, or change in metabolic activity, or change in local distribution of its members leads to the concept of dysbiosis, so an imbalanced intestinal microbial community. And this can be enhanced by diet, can be enhanced by drugs, antibiotics for example, can be enhanced by immune dysfunction. And in cirrhosis, the group of Jazz has nicely shown that changes in gut microbiota accompany cirrhosis and become more severe with decompensation. So if we go into the barrier aspect, then in fact if we dissect the barrier, and I'm going to go very briefly because I think Professor Abios will cover this topic somewhat more in extension. There are 4 elements in your intestinal barrier, which is in fact a physico-chemical barrier. So there's the luminal side, with bile having its biological effects, as you can see here. There is the mucus component, which is in fact composed of an outer mucus layer, which hosts the mucosa-associated microbiome. And then the inner mucus is in fact trapping the secretory antibacterial proteins, which immediately lead us to the third component, the secretory component, which is in fact comprised by the Paneth cells, which produce all of these nice antimicrobial peptides. And then finally there's the intestinal integrity. So the monolayer of epithelial cells is further enhanced by in fact tight junction proteins which act as a kind of seal. There are pore opening and pore closing proteins, occludins, claudins, zonular occludins. I will not go in depth. There are some nice reviews that cover this topic in recent papers. But what you should remember is in fact that there is a kind of multimodality line of defense with a whole army sitting behind a barrier ready to engage at each point that we are aggressed, or should wait if the aggressor is not to be expected an aggressor. In cirrhosis, each of these levels can be compromised, and what is in fact of importance is that if one layer fails, it does not necessarily lead to pathology, and it's probably rather the combination of different deficient layers that will lead to a chaos field in your intestinal wall. What then about immune responses? I will go briefly also on this because I think that Professor Abios will also cover this somewhat more in depth. So in fact the primary or the first line of defense is our innate immunity, which in fact detect invading bacteria by recognizing a highly conservative bacterial motif that is called the molecular or the microbiome associated molecular pattern. These are recognized by, in fact, pattern recognition receptors. Amongst other toll-like receptor 4 and 9 are of considerable importance. And in fact, once these are recognized either by tentacles of dendritic cells that hang out in the mucus layer, or by pattern recognition receptors on the epithelial cells, or by, in fact, transcytosis of microbiota through the layer by M cells that offer them to, in fact, dendritic cells subepithelial, the innate immune response will lead to, in fact, cytokine production through NF-kappa beta pathways, preferably. There are other pathways as well, but these, in fact, lead to, in fact, recruitment of immune cells, will lead to maturing and recruiting dendritic cells and antigen presenting cells, which in fact will further shape the adaptive immune response towards either further enhancement or shutting down. In cirrhosis, again, there are limited data, and there are many questions which remain to be solved, like how in cirrhosis bacteria are sampled, subsets of dendritic cells, the role of B and T cells, but I will not go further in this topic. I would rather switch to, in fact, the role of FXR as a potential master switch in gut-liver axis failure, as it has become one of our interests. So what is FXR? It's in fact a nuclear receptor that sensors for a broad range of natural and synthetic ligands, and it regulates a pleiad of functions, amongst others, so key hepatic functions, bile, acid metabolism, lipid, glucose metabolism. And with regard to the bile acid synthesis, it's controlled by negative feedback, while most of the other functions are governed by positive feedback, amongst others, so anti-inflammatory, vascular and barrier homeostasis. These receptors are highly expressed in liver, kidney and intestine, which are all bile acid handling organisms. And in fact, from this perspective, you could in fact put the FXR agent in the middle of what composes the gut microbiota host interaction at the most basic level. So of course, the first thing you want to know is that if there is a deficient gut-liver axis, if there is FXR deficiency. And there are experimental data, and there is a human receptor polymorphism study that in fact shows that in knockout mice, for example, that undergo bile duct ligation, that the number of bacterial translocation goes up by 10 times in comparison to wild-type BDL. And in humans, there is an association to SBP in patients with cirrhosis and ascites for a certain allele. In the next couple of slides, I will show you some of our experimental data that are currently resubmitted. And in fact, what we showed is that in BDL cirrhotic animals, both in the jejunum and the ileum, the FXR pathway is deficient. So we show SHP, which is one of the downstream effectors of the SHP, of the FXR pathway. I apologize. And then, in fact, what we did was try to revive that pathway by using an agonist obeticholic acid, or INT-747, which is a molecule that is currently in preclinical studies. So what we showed was that, in fact, in the jejunum, we did not get a reactivation, whereas in the ileum we did, which is to be expected if you know that bile acids are, in fact, reabsorbed in the terminal ileum. More of interest is, of course, that agonism of FXR by INT-747 decreases the number of bacterial translocation in treated animals compared to BDL with vehicle, and moreover, UDCA did not have an effect on bacterial translocation. And this correlated also with, in fact, SBP. In BDL, there was a high number of SBP, whereas in INT-747 treated animals, there was no bacterial translocation. What then about the effects on the intestinal barrier? So for this we went to in vitro assaying permeability by use of an Ussing chamber, in which we measured the transepithelial resistance. So this is in fact the resistance to passage of ions. What you can see is the jejunum and the ileum. So normal rats show high resistance. BDL rats show lower resistance, which implies permeability. And what you see again is that there is a selective increase in TER, so decreasing permeability upon treatment with an FXR agonist. If we correlate this with, in fact, the tight junction protein expression, and you look at the ileum and the jejunum, what you will see is that pore closing proteins do not change in the BDL versus control, whereas pore openers are highly expressed in BDL and remain expressed in the INT-747 groups, but in fact the pore closers, Claudin 1, are highly graded in INT-747 treated animals, which might explain the gain in or the decrease in the increase in barrier constitution. What then about immune responses? So here also we looked at mesenteric lymph node of our BDL treated rats, and what we in fact showed is that the high blood cell or the white blood cell count was increased in BDL rats, which was in fact mainly due to natural killer cells. And again, upon treatment with an FXR agonist, both were decreased, suggesting that FXR agonism controls immune regulation in the mesenteric lymph nodes. And moreover, if we look at the layer, the epithelial layer, most of the FXR targets were confined to immune cells. If we then look to cytokines, we saw that in fact interferon gamma was highly expressed in BDL, which was decreased again upon FXR agonism, and interferon gamma is one of the major cytokines of natural killer cells, so that makes the circle round. But how can these immunological findings and in fact permeability be combined? Well, for this we did in fact an experiment in vitro using a transwell setup. So what you do is in fact you take an insert which is covered with Kako cells. These are intestinal cells. These form a monolayer, and what you can in fact reproduce is in fact the passage of bacteria from the luminal side to in fact the distal side. And what we saw is in fact that in control there was limited amount of translocation of E. coli, which upon interferon gamma increased dramatically and could not be affected with INT-747. And this made us in fact conclude that intestinal FXR deficiency drives pathological BT indirectly through increased interferon gamma induced hyperpermeability, which can be in fact compensated by FXR agonism. Then I'm almost finished, Mr. Chairman. My gut microbiota are speeding up. So with regard to gut microbiota, I don't have any data to show you about the effect of FXR agonist on gut microbiota. These are currently underway. But what we did maybe as a first illustration is that in vitro, at least, we did a kind of susceptibility testing as you do for antibiotics. And in fact, we found no inhibition with increasing doses of the FXR agonist to all known pathogens available for in vitro testing. What then about clinical applicability? So this is only one slide. So there are some molecules that might prove of interest in the sense that there is the FXR agonist obeticholic acid, which is currently being tested in PBC. The results have been presented at ASLD and at EASL. So this is certainly a molecule which we could use for improving gut barrier function, and hopefully all that comes after it. On the other hand, there is also conicasterol, which is in fact SHP sparing FXR modulator with also PXR agonist activity, but this is mainly animal work and in vitro work. And maybe of interest for you, for those of you that have already test their or improved their FXR abilities, there are some suggestions that drinking coffee 3 cups a day for at least 3 weeks might enhance your FXR activity. There's also an illusion of red wine, and it's from a group from Malaga, Agustin. So they compared, I must say it was a very interesting design, a crossover randomized design. Either you drink gin or you drink red wine, and what you for 3 weeks. So I mean, it's an interesting concept. And what they found was in fact that the red wine drinkers had an increase in bifidobacteria, which might be of benefit for your gut microbiota. But I put a question mark because I don't want you to go home that you are allowed to drink red wine infinitely. So, but it might be at least a target. And then some conclusions, Mr. Chairman, ladies and gentlemen. So I hope that I have convinced you that gut microbiota and local intestinal host interactions forms a solid basis for a healthy gut-liver axis, and that it represents a complex interactive phenomenon. That FXR might be a potential master switch, because I don't think it's the only master switch. There are certainly others, depending on the etiology of cirrhosis, because I didn't allude to that, but different etiologies lead to different gut microbiota, and this might be a reason for some conflicting results. And on the other hand, there might be a clinical step up with the coming FXR agonists that we are already using in other contexts. And this is, of course, a major area of basic and clinical research in the future. But I have to agree with Voltaire at this point that, in fact, we are men or women who prescribe medicine about which they know little to cure diseases of which they know less, in human beings of whom they know nothing. But I'm sure this will change in the future. Thank you.